here, you people, you have no chalkboard.
Oli clear? Walieni? Tandika.
I'm clear.
Oyagala pose oyitaka pose.
Kano kenyeka bwento.
So oyagala wanika gundi ifyo
But it's saying pause.
Awo kigamba nti olaba time weba etambula wano. Wategeerera oli mu kusomesa. You just hello, welcome. You can talk about microbiology. I mean,
okay, fine. Tilt it a bit this way. Yeah, like that. This way. It's okay. Hello students, good afternoon. You are welcome for this lecture. We are going to talk about microbiology. Microbiology refers to the study of microorganisms, including bacteria, viruses, fungi, and parasites. So where do we find these bacteria, the viruses, fungi, and the parasites? These microorganisms can be found in the environment that is within the earth. Some of them can be found in the soil, some of them are found in the water, and some of them can also be found on the human body. Let's look at the benefits of microorganisms. When we talk of microorganisms, some of them are beneficial to the human beings, whereas some may not be beneficial. Let's look at the ones that are of benefit to the human beings. For example, we have bread production, or when they are preparing bread, we use some of these microorganisms. For example, the yeast, which we classified as a microorganism. When they are brewing alcohol, when they are producing the beers, they use also the yeast. When they are preparing cheese and the yogurt production, they also use some of the microorganisms. Preparation of antibiotics and vaccines, some of the microorganisms are being used. Then biological control, we use some microorganisms. Then production of gases, we find that some of the microorganisms are used for producing what? Gases. Let's go back to the history of microbiology. In ancient times or long time ago, the first recorded observations of microorganisms were seen in ancient Egypt and also Greece. During that time, people observed the effect of yeast and mold on bread and also wine. Then in around 1665, there was a Dutch scientist whose name was Antonio van Leeuwenhoek. He used a single microscope to observe some of the bacteria, and the microscope facilitated him to describe some of the bacteria and other microorganisms for the first time. So you can see that indeed microorganisms are very small. That's why he had to use that single lens microscope in order to look at what the bacteria looked like for the first time. Then around 1676, there was an Italian scientist. His name was Francesco Redi. For him, he conducted an experiment, and he later found out a theory of spontaneous generation. With this theory, which held that, or it stated that, living organisms could arise spontaneously from non-living matter. Okay, for example, living organisms could arise from what? Non-living matter. You very well know that at times matter is living and at times matter is non-living. Okay, so from the non-living matter, meaning that matter which has no life, some of the microorganisms arose from there. Then in 1796, there was also another scientist. His name was Edward Jenner. Edward Jenner developed the first vaccine for smallpox, and this smallpox vaccine was developed from a cowpox virus. Smallpox was a deadly disease in other years back, which was later on eradicated with the help of the smallpox vaccine. Then in 1857, there was also another scientist whose name was known as Louis Pasteur. Louis Pasteur, he demonstrated or he found out that there were some microorganisms that were responsible for the fermentation of wine and also beer. Okay, so because these microorganisms were responsible for fermentation of wine and beer, and it meant that there are some foods that we needed to stay for long so that we can use them. Then later on, he developed the pasteurization process. Pasteurization is a process of heating some liquids to kill harmful microorganisms. For example, the milk that is stored in the packets, that milk can stay for quite some days. Some can even stay for months. That is put in the packaged milk packets. At times you can see it's written on the packets, pasteurized milk. It means that that milk underwent a process of pasteurization whereby some of the microorganisms were rendered, where some of the microorganisms were killed. That's why that milk is able to stay for quite a long time, then we can be able to use it. So that was the process of pasteurization, which was developed by Louis Pasteur. Then in 1876, there was also another scientist. His name was Robert Koch. Robert Koch developed a series of postulates, and still this series of postulates have been used today. He proved that there were certain microorganisms that were causing certain particular diseases. As you are aware, there are a lot of diseases, but for Robert Koch, he postulated or he put forward that there were some particular microorganisms that were responsible for causing specific diseases. Then in 1928, Alexander Fleming Alexander Fleming discovered the first antibiotic, and the first antibiotic he discovered is known as penicillin. As you are aware, penicillin is used for treatment of a lot of diseases. This was discovered by Alexander Fleming. So penicillin treats most of the bacterial infections. Then in 1957, also another scientist by names of James Watson and Francis Crick, they published a paper describing the structure of DNA, and the DNA stands for deoxyribonucleic acid. So the DNA, when they studied it, it laid the foundation of the field of molecular biology. Most of the molecular biology is based on studies concerning the structure of the DNA. So, this now brings us to the slide on the classification of microorganisms. Microorganisms can be classified or they can be put into several groups or categories, and this is based on certain characteristics. For example, we are going to classify these microorganisms according to the size, the shape, the structure, and also the function. So, the main groups of microorganisms include the following. We have the bacteria, we have the viruses, we have the fungi, we have the protozoa, and also the algae. When you look at that diagram, on top, the box which is on top is written on microorganisms, followed by an arrow down. These microorganisms have been classified into the acellular. When we talk of acellular microorganisms, an example is the viruses. Then on the other side of the arrow, we have cellular. Then under the cellular, we have an arrow pointing downwards, whereby the cellular microorganisms have been classified into the prokaryotic. An example of prokaryotic microorganism, we have the bacteria, we have the archaea. Then under the cellular still, we have the eukaryotic. An example of the eukaryotic microorganisms, we have the algae, we have the protozoa, then we have the fungi. So, when we look at the divisions, the prokaryotic and the eukaryotic cells, we are going to look at their differences and similarities. For example, the prokaryotic cells, these are very small and they are simple. They range from 0.1 to 5 to 5.0 micro units in size. Then when you look at the eukaryotic cells, for them they are larger and they are a little bit more complex. So their size is 10 to 100 unimicrons in size. Then when you come back to the prokaryotic cells, they are unicellular, meaning that they have one cell and they don't have a nucleus, but however, they have a circular DNA, meaning that they have a circular deoxyribonucleic acid in their cell, whereas the eukaryotic cells, these are unicellular also, and they are unicellular, or at times they can be multicellular. For them, they have a nucleus which is present in their cell, and they have a linear deoxyribonucleic acid. So when you look at the prokaryotic cells, they have a single haploid chromosome, whereas the eukaryotic cells, they have a paired diploid chromosome. Okay, they have a paired diploid chromosome. That's why we are using the word diploid, whereas on the prokaryotic cells, we are using single haploid. That's why we are using the word haploid, meaning half. Then the prokaryotic cells lack a membrane. They lack a membrane-bound organisms. It means that the organelles that are found within the prokaryotic cells, they do not have a membrane. Whereas in the eukaryotic cells, the organisms are bound by a certain kind of membrane. Then, when we come to the aspect of reproduction, the prokaryotic cells produce sexually and also asexually. Then the eukaryotic cells mostly reproduce sexually. Then when you come to cell division, the prokaryotic cells produce both. Cell division is by binary fusion. Then the eukaryotic cells, the cell division is mostly by mitosis. Examples of prokaryotic cells include some bacteria, the archaea cells, and the examples of eukaryotic cells include the plant cells and also the animal cells. However, there are similarities between the prokaryotic cells and also eukaryotic cells. Some of these similarities include the following. You will find out that all the prokaryotic cells and eukaryotic cells, they have a cell plasma membrane. Then both the eukaryotic and prokaryotic cells, they have a cytoplasm. Prokaryotic and eukaryotic cells, they have a ribosomes. Then both of them have a DNA, that is deoxyribonucleic acid. Coming to the characteristics of microorganisms in terms of shape, microorganisms have various shapes. Some of them are spherical, meaning that they are shaped like a sphere. Some of them are rod-shaped, meaning that they are shaped like a rod. Then others are spiral-shaped, meaning that they are shaped like a spiral. Okay, and they also exist as single cells or aggregates. So these microorganisms also use various energy sources. For example, they need energy to perform their metabolic pathways or to perform chemical activities that are occurring within themselves. Some of these chemical pathways include the following. For example, the process of photosynthesis, respiration, and also fermentation. All these processes utilize energy. Then you'll also find that some of the microorganisms can cause disease in humans and also animals, whereas the others are harmless and you find that they could be beneficial to the humans. So microorganisms can develop rapidly, they can grow rapidly, and they lead to production of new strains that are resistant to antibiotics or more virulent than the previous strains. Microorganisms are small living organisms that can only be seen under a microscope. By the way, you cannot look at a microorganism using your naked eyes. You need to use a microscope because the microscope will help to magnify those microorganisms that are very tiny. The microscope will enlarge them so that you can be able to observe them under the microscopes. You will be able to see the shape, the size, and at times also the structure. Let's look at one of the microorganisms, for example, bacteria. When we look at bacteria, bacteria is a single-celled organism, and most of these bacteria are found anywhere on the earth. They can be found on the soil, they can be found on various surfaces, they can also be found on the body of the human beings. So, bacteria can be classified. We can classify bacteria according to the shape, and also classify it according to the staining properties and also other characteristics. Okay, so bacteria can be divided into many groups. We have the gram positive and the gram negative. So bacteria, remember we said that bacteria, they are also known as prokaryotic cells. What does it mean? It means that they do not have a nucleus. They don't have a nucleus and also other membrane-bound organelles. When we talk of organelles, these are the organelles that are found inside the cell of what? Of the bacteria. So instead, their genetic material or the genetic material of the bacteria is contained in a single cellular chromosome. And this single cellular chromosome is located in the cytoplasm of the bacteria. Remember, within the bacteria, we have the what? The cytoplasm. And this is where the chromosomes are located. Let's look at the uses of bacteria. Remember, we said that some bacteria were useful, and we said that some bacteria were also harmful. So one of the use of bacteria is the production of antibiotics. Antibiotics like streptomycin, these ones have been prepared from bacteria. Enzymes, proteins, and vaccines, these are made through biotechnology. Okay, for example, Escherichia coli, this is a kind of bacteria which has been utilized to produce human insulin. You've had some people who are diabetics, they inject themselves with the insulin. So some of these insulins are prepared from Escherichia coli bacteria. Then when it comes to the field of agriculture, in the field of agriculture, we have the rhizobium bacteria. Rhizobium bacteria, these form a symbiotic relationship with the legume plants, fixing atmospheric nitrogen and providing it to the plant as a nutrient. You remember, we have some of the leguminous plants, like for example, the beans, we have the cowpeas. When you look at the roots of the beans or the cowpeas, if you look at them, you will observe certain round structures on their what? On their roots. So those plants have the rhizobium bacteria, and we said that rhizobium bacteria form a symbiotic relationship with the plants in that they fix the nitrogen from the atmosphere, then these plants take in this nitrogen into them, and it forms part of what? Part of the nutrient. Then we have biomeditation, biomeditation of contaminated soil and water. For example, we have some other bacteria known as the Pseudomonas bacteria. The Pseudomonas bacteria breaks down the hydrocarbons in the oil spills, helping restore the affected ecosystem. Remember, in the soil, we also have what? Bacteria. Then remember, in the soil, we also could be having some spills from the oils. So the spills in the oils, remember the oil has hydrocarbons. So this bacteria known as the Pseudomonas bacteria, it breaks down the hydrocarbons in the soil, spills. Then when the hydrocarbons are broken down, then it means that the ecosystem will now be restored back to normal. So we also have another kind of bacteria which is used in production of various foods and also beverages. For example, the yogurt, the cheese, and the beer. Mostly they use the yeast. Then we also have another example. For example, the lactobacillus bacteria. This one is used to ferment milk and also produce yogurt and also cheese. The cheese that you have ever drank, the yogurt that you have ever eaten, remember, yeast has been produced and cheese has been produced and the yogurt is prepared, and we enjoy, most of us, drinking the yogurt and eating the cheese. This is because of the lactobacillus bacteria that are helpful to human beings. Let's move on to the structure of bacteria. Remember, we said that bacteria is made of a cell, and the bacterial cells, they also have different structures, and these different structures that are found within the bacterial cell, they are responsible for performing specific functions. For example, parts of the bacterial cell include the following. On the outermost part of the cell, the bacterial cell, we have a capsule followed by a cell wall. Then we have the cytoplasmic membrane. Then we have the ribosomes. Then we have the cytoplasm. Then we have the nucleus. Inside the nucleus is enclosed the DNA, or deoxyribonucleic acid. Then we have the pili. Then we have the plasmid. Then we have a tail-like structure which is known as a flagellum. Then we have inclusion bodies, and those are the structures that make up the bacteria. So let's discuss into details the different structures of the bacterial cell. Number one, we have the cell wall. The cell wall surrounds the bacterial cell, and because it surrounds it, therefore it provides it support. In other words, the cell wall supports the structures that are found inside the bacterial cell. The cell wall is also made of a protein. Cell wall is made of a protein, and it is made of a peptidoglycan. Peptidoglycan is a unique macromolecule. This macromolecule is only found in bacteria. Then we have a cell membrane. Cell membrane is a thin, flexible barrier. The cell membrane separates the cytoplasm from the external environment, and the cell membrane is made of phospholipids, proteins, and this cell membrane is selectively permeable, meaning that it will allow certain molecules to pass, and it will not allow some other molecules to pass, meaning that it will block them. That is why we are told saying that the cell membrane is selectively permeable, meaning that it will allow other substances in, then it will not allow other substances to enter the bacterial cell. Then we have the cytoplasm. The cytoplasm part is a fluid, meaning that there is a fluid inside it, okay? So this fluid-filled interior of the bacterial cell, it's the one which is having the genetic material, okay? It contains the genetic material. In other words, the material which makes this bacteria to continue producing more and more and more bacteria, okay? So it contains the genetic material, it has the ribosomes, and also enzymes, and also metabolic pathways that are important for the cell's survival. Remember, for the cell to survive, it means that this cell has to reproduce, okay? And for this cell to reproduce, it also means that this cell is responsible for carrying out metabolic chemical reactions that take place within this cell, such that this cell can be able to grow, this cell can also be able to reproduce and continue surviving. Then we have another structure which is long and it looks like a tail. They call it flagella. Okay. Flagella, these are long whip-like structures. They extend from the bacterial cell wall. Okay. The flagella helps the bacterial cell to move. Okay. It helps the bacterial cell to move, and also helps the bacteria to move towards its nutrients or away from what? Toxins. For example, maybe if there are some poisonous substances that are near it and it feels they will harm it, then this cell will help the bacteria to swim away very fast. Then we have another structure which is known as the pili. Pili looks like a hair, that's why we are saying pili are hair-like structures. These hair-like structures protrude, or they are seen from the surface of the bacterial cell. And what is its function? The function of these hair-like structures is for attachment to surfaces and also for the transfer of the genetic material between the bacterial cells. Remember, we said that bacteria also produce. So if the bacteria are also to divide, it means that some of this genetic material has to be transferred to another cell. Then we have the plasmids. These plasmids are extra chromosomal. These are extra chromosomal DNA, and these are usually present in multiple copies. In other words, they also aid in cell division. Then we have the endospores. Endospores, these are a dormant form of the bacterial cells. These dormant cells of the bacterial cells, these are produced by certain bacteria when starved. Okay? When starved, it means that the bacteria is dormant, it's lying dormant, okay? That's why we say that the bacterial cell have been starved. So the actively growing state of the cells is referred to as vegetative. So here it means that the spores resist adverse conditions, including high temperatures and organic solvents. In other words, what does it mean? When we talk of endospores, it means that the bacterial cells that have been produced by certain bacteria These bacteria cells are resistant. They are resistant to high temperatures and even organic solvents. What does it mean? It means that when these cells are exposed to very high temperatures, the bacteria will not die. Even if you expose these cells to certain other solvents, maybe in hope of maybe killing them, these bacterial cells may not be able to. These bacterial cells will still stay alive. They can survive unless if we use other means like maybe very, very higher temperatures, like maybe in the autoclave, then these bacterial cells can be destroyed. Remember, we said bacteria can also be classified into different groups based on various characteristics such as their shape, their size, their metabolic activity, and also genetic makeup. When we classify bacteria according to their shape, we have the cocci. The cocci, these are spherical. They are shaped like a sphere. That's why we call them spherical. Then under the cocci group, we have the bacilli. Then we have the rod-shaped bacteria. An example of rod-shaped bacteria. Some bacteria are rod-shaped. Sorry, we have the bacilli. The bacilli, these are rod-shaped. Some of the examples, we have the staphylococci. We have the coccobacilli. Then we have the diplococci. An example of it is the Vibrio. Vibrio causes cholera. Then we have the spirilla or the spiral-shaped bacteria. Examples of this, we have the streptococci, we have the coroniform bacilli. Then we can also classify bacteria according to a certain method which is known as gram staining. Bacteria can be classified based on the response to certain other staining techniques, which we call a gram stain. So this gram stain will help to differentiate this bacteria into two main groups. For example, we have the gram positive and gram negative bacteria. When we talk of gram staining, when we talk of gram staining, gram staining. When we talk of gram staining is a technique. This is a technique which is mostly used in the laboratory. Okay, so this technique differentiates bacteria into two groups or two categories. And these two categories are based on their cell structure. For example, we have the gram positive and gram negative. However, this technique was discovered by a Danish bacteriologist named Hans Christian Gram. He developed this technique in 1884. So the gram-positive bacteria, they have a thick layer of peptoglycan in their cell wall, which makes them actually stain purple or blue when they are exposed to crystal violet stain and iodine solution. Whereas the gram-negative bacteria, remember we said that this bacteria have a thinner layer of peptoglycan peptidoglycan and an outer membrane that has lipopolysaccharides. So when these bacteria are stained, when these bacteria are stained, the stain will make them stain pink or red when they are exposed to counter stain such as saffron. Let's look at the gram staining techniques or the gram staining technique. Suppose maybe one day you are in the laboratory and you wanted maybe to do a gram staining of bacteria. Step number one, the bacterial cells need to be fixed to a microscope slide using heat. Then after fixing the bacterial cells onto a microscope slide using heat, the second step is the slide will be floated with the crystal violet stain, which is left to sit for 1 minute. Then thirdly, the slide will be rinsed with water to remove the excess stain in case you had put a lot of stain on it. And then this slide will be floated with the iodine solution, which binds the crystal violet to the cell wall. Number 4, then the slide is rinsed again with water and then exposed to a decolorizing solution such as alcohol or acetone. This will remove the stain from the gram-negative bacteria's cell wall. Then later, the slide is rinsed with water and then stained with a counter stain such as suffranin, which stains the cell wall of the gram-negative bacteria. So there, you'll be able to tell whether the bacteria is gram-negative or gram-positive according to the stain that it will have picked up. Metabolic activity. Bacteria can also be classified based on metabolic activity. For example, you will find that there are certain kinds of bacteria that require oxygen for growth. And this kind of bacteria are known as aerobic bacteria because they require oxygen for growth. Then you will find that there are also certain other kind of bacteria which do not require oxygen for their growth. So because they don't require oxygen for their growth, we call them anaerobic bacteria. Then we also have the facultative type. The facultative type can grow with both oxygen or without oxygen. For them, they can grow. DNA sequence polygenetics, comparing the genetic sequences. Bacteria can also be classified when we are comparing the genetic sequences, their DNA sequences, the polygenetics, comparing to the genetic sequences. They look basically at the sequence of the genes of the bacteria. Then we have the genomic characteristics. When we talk of genomic characteristics, we are looking basically at the size, the complexity, the presence or absence of specific genes. For example, the ribosomal, that is RNA, the ribosomal sequence is a fundamental component of the bacterial ribosome. These sequences can also be looked at. Then the protein profiles or protein patterns, these are also able to distinguish certain kinds of particular bacteria species. Bacteria can also be classified according to bacterial pathogenicity. When we talk of bacterial pathogenicity, it refers to the ability of the bacteria to cause disease. This disease can be caused in human beings, animals, and also plants. Bacteria sorry. Bacterial pathogens have also evolved various mechanisms to survive and thrive in different hosts. Okay? Bacteria have also defined their own ways such that they can be able to survive and continue growing either in the humans or in the animals and also maybe also in the plants. So these mechanisms that have evolved enable the bacteria to survive and thrive in different hosts, including invading the immune system, invading the colonizing host tissues, and also producing toxins that damage the host cells and also tissues. What do we mean by this? What we mean by this is that certain other bacteria, when they invade the host or when they invade the plant or the animals, they develop means of surviving there such that they can continue to damage the cells or the tissues of the human or the plants. Bacterial pathogenicity, we said that referred to the ability of bacteria to cause disease in humans, animals, and plants. Bacterial pathogens, we said that they have evolved various mechanisms to survive and also thrive in different hosts. Then they will continue invading the immune system of the individual. They will continue to grow in the cells. Then they will continue to produce toxins as they continue growing. Then they will end up damaging the cells of the host or the plant. Let's continue with the bacterial pathogenic factors. There are some bacterial pathogenic factors. When we talk of pathogenic factors, these are the various characteristics and the mechanisms that bacteria use to cause disease in a host organism. For example, we said that bacterial pathogens have developed multiple pathogenic factors that enable them to survive and multiply in host tissues and cause damage to host cells and organisms. What do we mean by this? It means that these bacteria have developed tricks. They have developed their own tricks, to put it in the simplest ways, where the bacteria have developed their tricks, of which these tricks will enable them to continue growing, continue multiplying, so that they can be able to damage or to harm the cells of the host or the organism. One of the bacterial pathogenic factors include the following. One, adhesion. Okay, adhesion, these are surface proteins that are known as adhesins. These adhesins allow the bacteria to bind to specific receptors on the host cell. When you look at the cell of the host, it could be having some specific receptors, okay? So these adhesins allow These adhesins allow them to bind on specific receptors on the host cells. These receptors could act as maybe receivers that are found on the what? On the host cells. So these adhesins enable the bacteria to colonize and infect the host tissue. In other words, these adhesins will allow this bacteria to attach onto the host tissue and invade it and cause harm or damage to the host cells. Then we have invasion. Once bacteria have adhered to the host cell, they use a variety of mechanisms to invade the host cell, including the production of enzymes that degrade the extracellular matrix and the use of specialized secretion systems that inject the bacterial proteins into the host cells. What do we mean by this? Remember, we said that once these bacteria have got stuck on the host cells, they will use a lot of tricks or they will use a lot of mechanisms to invade the host tissue. Okay? They will invade the host tissue, and when they do so, they will produce enzymes. When these enzymes are produced, these enzymes will degrade the extracellular matrix. When we talk of the extracellular matrix, this is the matrix that is found within the what? Within the host cell. So they will degrade the extracellular matrix and use it and the use of specialized secretion systems that inject bacterial proteins into the what? Into the host cell. In other words, when they degrade the extracellular matrix, then they will use other systems or other secretion systems to inject the bacterial protein into the host cell. Toxins. Bacterial toxins, these are proteins that cause damage to the host cells and also tissues. Or toxins are poisonous proteins, okay? Toxins can mean poisonous proteins. Bacterial toxins are proteins that cause damage to the host cells and tissues. Or you can call bacterial toxins poisons, to put it in the simplest way. So there are two kinds of bacterial toxins. The first one is known as endotoxins. Endotoxins are part of the bacterial cell wall. For them, they cause systemic effects. For example, they can cause an individual to have a fever, they can cause sepsis, they can cause exotoxins. These are secreted by bacteria and they can also cause effects on the host cells. What effects will they cause on the host cells? For example, they can cause destruction of the red blood cells. They can also inhibit the protein synthesis, meaning that there will be no more synthesis of proteins, and they can also destroy the cell membranes or disorganize the cell membranes. Then we have the immune evasion. Bacterial pathogens have evolved various mechanisms to evade the host immune system. For example, the immune system of the individual will be invaded by this bacteria. Okay, this will include the use of antigenic variation to evade recognition of antibodies and also the production of proteins that inhibit complement activation and the modulation of the host cytokine and also chemokine responses. So this immune evasion is a little bit complex, but to put it in simple ways, it means that the bacteria pathogens, remember we said that they developed different mechanisms for invading the host immune response. Okay? They developed various mechanisms for invading the host immune response, whereby it includes the use of antigenic variation to evade recognition by antibodies. In other words, the antibodies of the host will not be uh, including the use of antigenic variation to evade the recognition of antibodies and the production of proteins to inhibit complement activation and also modulation of host cytokine and also chemokinin responses. So here it means that the immune system is the one which is invaded. Okay, so when it is invaded, then at times there may be difficulties for the antibodies to there may be difficulties for the antibodies there may be difficulties for the complement activation and modulation of the host cytokine and chemokine responses. Then we have quorum sensing. Quorum sensing, here some of the bacteria use quorum. Quorum sensing is a cell-to-cell communication system. What does it mean? It means that the cell-to-cell will coordinate their virulence factors and also regulate their behavior. So this enables the bacteria time. This gives the bacteria to time their infection for maximum effect. In other words, the cell-to-cell communication, it means that these cells are communicating together. They are coordinating together. They are coordinating their virulence factors and they regulate their behavior such that they enable the bacteria to take time so that it can be able to exert its effect of infection. Thank you for listening. Have a blessed day.